So today I'm going to be talking to you guys about the public health implications of a disease called river blindness. And this is a vector-transmitted disease, and the causative agent is the filarial nematode Onchocerca volvulus. Now many of you are obviously familiar with the epidemiologic triangle model of cyclic disease transmission, as seen in Dr. Castle's presentation earlier. However, there are many complex internal and external influences that make management of river blindness a lot more complicated and do not necessarily conform to the simplicity of this model. And I'm going to be looking at the transmission cycle and symptoms, the vector and parasite control programs, and the progress that has been made towards the elimination of this disease. Now, river blindness, the medical designation of this disease is onchocerciasis, and it's classified as a neglected tropical disease. And now this means that it typically affects disadvantaged individuals in isolated and rural areas, and control and elimination programs have been traditionally underfunded. Now, onchocerciasis stands as the second leading cause of infectious blindness worldwide. It's estimated that around 120 million people are currently at risk for infection, 18 million people are infected, and there are around 40,000 new cases of blindness that occur each year. There are vast social implications for this disease. It greatly disrupts economic development. There are implications for community structures, and it also influences the livelihood of younger generations because children are often forced to stay home from school and take care of older generations who may be blind or suffering from severe symptoms. And there are elimination goals in Latin America for the year 2015 and in Africa in the year 2020. Now, the geographic distribution, it's mainly found in Africa and Latin America, although there are some small endemic populations in the Saudi Arabian Peninsula and Yemen. However, 99% of the caseload of onchocerciasis is found in Africa. And lotic, non-static bodies of water are important habitats for the vectors of this disease. And so this makes populations that live by rivers or other moving bodies of water more highly susceptible to infection. And what makes the spread of onchocerciasis different between these two continents is the distribution of endemic populations. In Africa, it occurs countrywide, and you can see in Latin America, these red regions here, it only occurs in concentrated foci. So Onchocerca volvulus, this is the parasite responsible for the manifestation of dermal and ocular symptoms. There are two main stages of this filarial parasite, the macrofilarial stage, which is the adult stage, and the microfilarial stage, which is the larval stage. The macrofilarial worms can grow up to 2 feet in length, so these are pretty nasty, and they can live between 10 and 15 years in the human host, and this necessitates very long-term treatment. Now, the macrofilarial worms live in adult masses in the subcutaneous nodules. As you can see here, these are called onchocercomas. This is actually generated by an immune response to these worms found in subcutaneous and muscular tissue. And also, what's important for Onchocerca is it also harbors a bacterial endosymbiont called Wolbachia, and this is important for parasite fecundity, survival, and it's proposed to exacerbate the pathology of this disease. And the microfilaria, they circulate in subcutaneous tissue. They do not enter your bloodstream at all. And it is the death of these microfilaria that trigger the immune reaction, which causes severe itching and skin depigmentation. So vector development, the sole vector of onchocerciasis is the black fly, and all vectors, or all stages of this vector's life cycle are aquatic, except the adult stage. And optimal development of the black fly larvae are dependent on dissolved oxygen content of the water body, temperature, they like colder temperatures, pH, the velocity of the water body, and the presence of dissolved organic matter, because these larvae are filter feeders. And larval density is positively correlated with the velocity of the water body. So more or higher densities of larvae are found in the highest velocity portions of a water body. And multiple bites are needed to contract symptoms of onchocerciasis. So the transmission cycle of onchocerciasis begins when a black fly takes a blood meal from an infected individual. Only the female counterpart takes a blood meal, and she does this before she lays her eggs. And by taking a blood meal, some of the microfilariae are taken up, and they migrate to the middle portion, the mid-gut of the black flies. You can see circled in red in this image. And they develop through 3 stages of molt in the muscular tissue. After this 3rd stage, they re-migrate back to the black fly's mouth, and once the black fly goes to take an additional blood meal, these microfilariae are transferred back into the human host. Once they enter the lesion and made by the black fly in the human host, they go through 2 more phases of molt, and that's when they become fully mature adult, either male or female, macrofilarial worms. And this is just an image summing up the transmission cycle, that there's 2 basically different cycles. One that happens in the black fly, which is an intermediate host, and an entire human stage, which is also mainly the reproductive stage of this disease. So there are many manifestations associated with this disease. Most fall into ocular or dermal categories, and the most common ocular manifestations are keratitis, and this is inflammation of your cornea. And you can also have uveitis, which is inflammation of the middle layer of your eye. You can have optical atrophy, which is the shrinkage of your optic nerve. And the most severe symptom associated with onchocerciasis in terms of microfilariae in your eye is total blindness. You can also have a wide variety of dermatologic symptoms. Intense itching, dermal nodules, as I mentioned before, skin depigmentation, which is also called leopard skin, which is because of this spotted formation, loss of skin elasticity, skin scaling and hardening, and Sauda in Yemen, which is basically the presentation of these symptoms on one limb. And you can also have symptoms that mimic lymphatic filariasis, which is enlargement of certain appendages. So control measures of this disease, it's been mainly a 2-pronged control approach for river blindness, either aquatic vector control or management of the parasite Onchocerca volvulus to help mitigate microfilarial load within individuals. And there's been 3 primary control programs: the OCP, which is the Onchocerciasis Control Program that ran from 1975 to 2002, the APOC, which was the African Program for Onchocerciasis Control, which began in 1995 and is still ongoing, and the OEPA, which is the Onchocerciasis Elimination Program for the Americas, which started in 1993 and is still ongoing as well. So for aquatic vector management, there's been 3 major stages. The first stage focused on removal of aquatic vegetation, both lining rivers and in the river, and this was mainly a strategy to remove habitat of black fly larvae and food sources because of the dissolved organic matter. However, this hasn't been very successful. The second stage was trial spraying of DDT in waterways, and this was somewhat successful in terms of larval eradication, but we also saw instances of fish kills, especially in Kenya. So there was also a need for the development of an additional strategy that had less toxic effects. So in 1975, this led to the development of the Onchocerciasis Control Program, which was mainly focused in West Africa. And this was a large-scale aerial larviciding control technique that focused originally on the use of timofos, which is an organic phosphate pesticide. And what makes this vector unique is the aquatic stage from egg to pupae is about a week, and this necessitates spraying of insecticide almost weekly. So within 6 years of this program, we began to see resistance in black fly species. So they implemented a rotational strategy with different insecticides, such as other organophosphates, carbamates, pyrethroids, and Bacillus thuringiensis. And by the end of the OCP, 7 different insecticides were in this rotation. And at the peak of larviciding activity between 1986 and 1991, around 50,000 kilometers of river were being treated, and this was in an area of over 1 million square kilometers. So, and you can imagine this is weekly. So very vast impacts. So obviously, having a larvicide campaign of this scale necessitated ecological monitoring. And the main group in charge of this was, I know not a very creative name, but it was termed the ecological group. And they were in charge of running toxicological tests to evaluate the effects of all these larvicides on aquatic habitats. And this was important not only to sustain ecological well-being, but also to ensure economic stability for the fishing industries that are very important to these local regions. And basically what they did was they had a specific criteria for the selection of larvicides, which was based off of seasonal hydrologic cycles and also the discharge rates of different water bodies. They mainly looked at fish and invertebrate species, and the main effects seen from this was in invertebrate species was the rarefication of some species and in some cases extinction. However, they did conduct resiliency studies, and it was shown that these invertebrate species could repopulate these areas. However, these results are inconclusive because they didn't really analyze how decreases in these populations affected trophic interactions in these regions. And in fish species, special attention was paid to acetylcholinesterase activity because in organophosphate pesticides, one of the main ways it causes toxicity in individuals is inhibition of this enzyme. And it was seen in some tilapia species that around 20% decrease in activity was seen, but all other population fluctuations were attributed to seasonal hydrology, and this was because during a lot of the monitoring in this program, a major drought occurred, so that also influenced the results of their studies. The second management strategy has focused on microfilarial management with the drug ivermectin, which is also used in animal species, as you can see, HeartGuard, which you give to your domestic dogs and cats. But this has been found to effectively kill the microfilarial species, and this treatment was produced in 1987 by Merck. And the main impediment with the use of this drug to treat is that it doesn't kill the macrofilaria worms. So they're still living inside the human host and are still able to reproduce. And this necessitates treatment with ivermectin for the entire lifespan of the macrofilaria worm, or even longer, which is 15 to 20 years with the use of this drug, which is pretty non-sustainable. And the use of mass drug administration programs has prompted the occurrence of resistance in some populations, and this is a big problem because we currently do not have a backup plan if this was to become more prevalent. In terms of treatment for macrofilaria worms, our techniques are very inadequate. We have nodulectomies, which is basically the surgical removal of the onchocercomas, the dermal nodules on the skin. But many macrofilaria worms live deeper in the muscular tissues and are inaccessible to this removal technique. Also, another medication called Suramin was also used in some instances, but this has proven to be too toxic for mass use, and it also causes optic nerve diseases and can cause blindness itself. So we really don't have an effective measure to take against these macrofilarial worms. Each of these programs has taken sort of a differential approach to control of this disease. The OCP mainly focused on aquatic vector control through this larviciding. In the OEPA, however, there's been more of a focus on ivermectin dosing. They do a twice-a-year regimen, and they've actually had 85% compliance with their endemic populations. And the reason for this is there's been less of an influence on aquatic vector control is because the populations with endemic onchocerciasis are much more isolated, and low-lying cloud cover in these areas sort of disrupts the effectiveness of aerial larvicide spraying, and it also they had worries about how it would affect non-target fauna species that are important for coffee pollination in these regions. In the APOC, this implements integrated vector management with mass ivermectin dosing programs and larvicide application when applicable. And in Yemen, there's no distinct elimination program for onchocerciasis. It's more passive distribution of drug, ivermectin. So elimination and feasibility. In Latin America, we've made great strides towards elimination of this disease. In 7 of the 13 endemic foci, they've actually been able to stop treatment with ivermectin and are on their way for certification of elimination of transmission. However, this success has not been seen in Africa, despite around over half a century of treatment with aquatic vector control and ivermectin drugs. We have seen suppression in blindness and alleviation of symptoms in some areas, but it definitely has not been all that successful. However, there's urgency to find a better elimination program because funding is slated to end for the APOC program in the year 2015. So that only gives us around 3 years to come up with a more sustainable solution. So future directions, development of an antibiotic to treat or kill the Wolbachia bacterial endosymbiont, a drug that kills the macrofilaria worm, which would allow treatment to be shorter, not between 15 and 20 years, which is sort of unsustainable. We need more stable community-based treatment, which can survive civil unrest, which is also a problem in African countries with onchocerciasis. We need increased surveillance of resistance, and we also need further studies on ecological larvicide use, how this has influenced reproductive capacity in fish. There's not been a lot of studies conducted on this. And so my conclusion of this study is basically we need to take a new approach to the elimination of onchocerciasis. Instead of focusing on control of this disease, we need to change our perspective and develop an elimination program for Africa so we can have a concrete, feasible goal that can be met. But with the resources we have now, the year 2020 might not be a feasible goal for elimination of this scale in Africa. So thank you for listening.